good morning, students. Hi, how are you? Okay, hope you're having a wonderful Monday. Okay, of course, you can't say wonderful Monday. Yeah? Everybody hates Monday, right? Okay, including myself. Okay, but doesn't matter. Okay, we all have to study, right? We have to work. Okay, so Joanne, good morning. Sophia, Ziling, Chia Pei Shan, okay, and everyone who's out there, all right, good morning to you all. Okay, so today I'm very excited about the lesson because, you know, this is one of my favorite topics, which is on genetics and inheritance. So today we will continue with what we started. My lesson was on Saturday, 2 days ago, where I gave you an introduction on all the terms that we need to use when we want to describe inheritance, okay, or the crossing of one organism with another organism to describe what is the next generation, the composition or the properties of the next generation. Okay, so we have already discussed some terms. Today we're going to learn how to draw a schematic diagram to show monohybrid inheritance. Okay, so here I see Pei Shan, Shino, Li Lin. Yes, good morning everyone. All right, now can I just share screen and let me know if you can see this? I think you can hear me, right? Okay, if not, please do give me let me know feedback. Okay, so this is what you're going to learn today. How to draw this diagram. Okay, what I mean is this diagram shows you what happens when you take one organism and you cross it with another organism. Let's say, of course, it must be same species, but different characteristic or different trait. Call it different trait. Same characteristic, but different trait. Let's say one tall and one short. Okay, so you have one individual that is tall, one individual that is short, or P-plan. In this case, it's easier for me to let's discuss what Mendel has done for experiments. With experiments, he uses P-plan as his subject, which is the Pisum sativum. So he took a tall plant and then he crossed with a short one, because in the P we call it the Pisum sativum species, there are actually very distinct two types. It's either tall or either short. There's no intermediate. There's no something like in the middle. Okay, so you will have either all the Pisum sativum is tall or tall, and then either whether it's tall or short. There's nothing in the middle. There's no intermediate characteristic. That's what we mean. So we use to tall and short. If we combine it, that means we interbreed between these two individuals. What is the next generation? What is the characteristic of the next generation? Are you going to get tall, or all short? Or is there going to be a combination of tall and short? And if there is, how many tall, how many short? Or is there going to be an intermediate? Are you going to have a medium height? Okay, so this is what fascinated Mendel, okay, more than 100 years ago. So he worked extensively. He also experimented extensively on this Pisum sativum. Okay, so today we're going to learn how to draw this diagram. All right, okay, so you can see this. Let me check out now. Yes, I can see it now. Right, you can see this now, hopefully. And not only tall and short, you can have this crossing. You can have, example, purple and white. So in Pisum sativum, you also have two types of flowers, two kinds of variant. You call it variant. You call it traits. Either purple flower or white flower. There are no other types, either purple or white. So when you crossbreed a purple plant, a purple flower plant with a white flower plant of Pisum sativum, both must be Pisum sativum now, of course. Are you going to get purple, or are you going to get white? Or are you going to get a combination of purple and white? Or are you going to get something in between? Okay, that means purple mixed with white. So that's what I mean. Okay, so before we look at the drawing, how to draw this, let me revise what you should already know. Okay, that's what I mentioned to you. Those who have not joined my class on Saturday, you should go and watch the first video, which is Monohybrid Inheritance part 1. Okay, part 1. Because without that, you will not be able to understand today's lesson. Okay, so here we are going to look at what we did last lesson was to learn about the different types of, we call it the alleles. Alleles are the variation of the gene. Okay, so you say, for example, you have characteristic is height. Okay, so height can be tall or short. So similarly, this one, there is a gene for height. Okay, that's what we mean by gene. There's a gene for height. And the different types of gene of height are called allele. So there is a tall allele and a short allele. So you make sure you know all the what you call the terms related to inheritance before you can understand the further lessons. Okay, so gene, it controls the characteristic and allele controls the trait. Okay, so gene is similar to like characteristic, and allele is the different type of gene that controls the different traits. So trait is either tall or short. Okay, so you have two types of alleles, which is dominant and recessive alleles. All right, so dominant we will use capital letter, and short we call recessive allele we use small letter. Okay, we also look at what is genotype and phenotype, and then homozygote and heterozygote. Okay, now let's go straight to today's lesson. If you're still not too sure, I suggest you have a look through again the previous lesson to understand this. So let me go through this one first. This is, I didn't show the answer to all this. So let's look at seed color. Let's say seed color phenotype is what is observable. The color is yellow. You can see from the outside. That's called phenotype. Okay, and the other type will be green. So this is traits. Okay, trait means the different variation of the characteristic. Okay, seed color is the character or characteristic. The trait is either yellow or green. And then in order to represent the trait, we need to use symbols. Okay, we call it the alleles. So for genotype yellow, let's say we use the symbol of, let's say B, B. So there are two types. Whether both the alleles are dominant, or it can be one dominant, one recessive, it will still be yellow. Okay, because the capital B is dominant. And then in order to show green color, seed color, it has to be two small ones. We call it two recessive alleles. Then only it shows green color. Okay, so here is all the same. So if you want to put, you can just fill up here. All right. So green, let's say you use capital G, and then or this one will also show green. For yellow pot color, let's say this one, double small letter G. Okay, let's go on to the next one. Now, these are what I've shown you last lesson, the definition of all the different terms. Okay, now let's look at monohybrid inheritance. This is today's lesson. And then in explaining monohybrid inheritance, we have to look at Mendel's first law. So Mendel devised his first law. He used this law to explain what his results. He always get the same result every time when he did experiment. And he has to be very smart, because if you are not able to you will not be able to explain it if you cannot understand what actually happens. So he's able to put it all into something that we can understand. Okay, and it's called the first law of inheritance. So we're going to look at it. Anything else? Nothing so far. All right, nothing in the chat. Okay, now, why did he use Pisum sativum as its object or subject of study? Why did he use Pisum sativum? Why can't he crossbreed animals? Or let's say rat and rat. Okay, male rat, white rat, and male that is, let's say, white rat, all right, and then cross with a female that is brown color. Okay, because rat are different color, right? We got albino, which is white, and you have the normal color, which is brown or which is black. Okay, why don't we use that? There are a few reasons why he didn't use animals, right? And then he preferred to use pea plant. Now these are the reasons. Number one, it's easy to grow. Okay, plants is the pea plant is easy to grow, right? And easy, of course, to handle compared to animals, right? And the life cycle is short. Now, I've Googled up, the life cycle is roughly about 60 days. That means when you start to plant your seed for the Pisum sativum, you just need 2 months before you can harvest, you can get the pot ready. The seed is ready for the next generation. So that means you just need to wait 2 months before you can actually have the next generation. So it takes so short, 2 months already, you can have the next generation already. But for, let's say, humans, if you want to study on humans, our life cycle is so long. Okay, when a baby is born, you have to wait for it to become, let's say, maybe 20 years old, for it to get married and then have children, and then only you can have the next generation to see the effect. You have to wait for a long time to see the inheritance in the next generation. That's why I mean you need 20 years at least, 10 to 15 to 20 years to have the next generation. So it's too long, isn't it? And some animals also is difficult to handle. So using plant is easier to handle. So life cycle is so short that you within 2 months you can have the next generation already. You can see the effect. Okay, the father, let's say the plant is a purple flower and you cross with a black flower. You breed it and then it will produce a fruit, you produce the seed. Within 2 months, you plant it again, you will see the next generation and see what is the color of the flower in the next generation. So it takes a short time to finish your investigation or your study. Okay, the pollination can be controlled easily, yes, because they have anther and ovary. You can see this picture here. And then the ovary, all right, is where you have the female gamete, right? The ovaries are here. And then you have the male gamete, it's in the anther. So if you want to prevent self-pollination, you do not want it to self-pollinate, what you do is you just cut off the filament to separate the anther, so it will not self-pollinate. Or if you want it to pollinate cross-pollination, that means you take the anther from another plant and then you brush it and put it onto the stigma of the female. Okay, it's very easy for you to handle. So this one you can actually do it yourself. Right? You take a brush, you take a pollen grain from a male plant and you brush it onto the stigma of the female, and then you will get you already induced pollination. So pollination can be controlled, whether you want to do self-pollination or cross-pollination. Okay, you can handle easily. Okay, next one have both male and female parts. And then next one, the traits of this plant can be studied easily. Now, okay, why easily? Because the traits are contrasting. They are very obvious. There are two types of traits. Example, purple flower and white flower. They are very obvious. There are only two types. You don't have so many different types, or you don't have what they call a mix of purple and white. You find it difficult to see when you look at the flower. Oh, is it purple or is it white? You're not sure. But for this one, it's very clear. It's either purple or it's either white. Okay, so there are 7 traits that Gregor Mendel investigated on, and those are the 7 traits here. And I show you these 7 characteristics here. Okay, seed shape, okay, whether smooth or wrinkled, so it's very distinct. The difference is very, very clear. Seed color is either yellow or green. There are no other types. Okay, the pod shape is either inflated or constricted. Okay, inflated means it's like blown up, like a balloon like that, right? And then the constricted is like kecut already lah, right? It's like shrunken. Okay, pod color is either the outside, the pod color is either green or yellow. It's also very clear. Flower color is purple or white. Flower position is either axial or terminal. Axial, terminal is at the end. The flower is at the end. Axial is in the branches, in between the branches. Okay, and the height, whether it is tall or dwarf. That's all. Very easy to see. So we call it contrasting traits. That's why he chose Pisum sativum. So basically, they have contrasting traits, and then it's also very easy to have the next cycle, the next generation. The life cycle is very short. Very easily, you can already finish your investigation in a short amount of time, and you can repeat as you need. Okay. So self-pollination, you've noticed that before in Form 3. Cross-pollination is where you take the pollen of one plant and then you put it on the stigma of another plant, not the same plant. That means the flower, the anther, the pollen grain from one flower, plant A, you put it onto plant B, the stigma. So like this, very simple. Let me draw this here. To show what is cross-pollination, it's not a repeat because I've done this in Form 3. So you have plant A, let's say, okay, I put tree lah. This is tree A, and this is tree B. Okay, the name of the tree. Okay, so A will have its flower. All right, many flowers. I can't. Okay, one plant can't be just having one flower. You have many flowers. Okay. Okay. Then B also has its own flower, right? Okay. Now, in order for it to be a self-pollination, let's start with self-pollination. Self-pollination means you take the anther of one flower, you put it to the stigma of another flower, you pollinate. Okay, it can be the same flower. This is self. That means there's one flowers is only one. All right, the pollen from this flower drop onto the stigma of this flower is self-pollination. Self-pollination. right? Same flower, one flower. Or it can also be from one flower to the next flower, but on the same plant. So the most important thing, the concept is it must be the same plant. Doesn't matter same flower or not same flower, it's still considered self-pollination. Okay, it's still considered self-pollination. That means different, let's say X here, this is flower X, this is flower Y, this is flower Z. X, the enter as a pollen grain from X, drop onto the stigma of the Y, another flower, but same plant. So most importantly, it's the same plant. It's still considered self-pollination. Now, where is it cross-pollination? Cross-pollination happens when you have the pollen grain of plant, let's say flower Y, it drops onto another flower, but it's not onto the same plant, different plant. Okay, so this will be considered as cross-pollination. It cannot be on the same plant, so it's got to be a different plant. Okay, of course, same species. We're not talking about crossbreeding different species. Same species, but it's another plant. It's considered cross-pollination. Okay, so the stigma, the pollen grain of one flower drop onto the stigma of another flower on another plant. Okay, so that's called cross. Understand? Right. So now, what he did was he took the pollen grain of a certain flower. If he wants to crossbreed, he just brush the pollen grain onto the stigma of whatever plant he wants to pollinate. He can control it easily. Okay, that's why he chose the Pisum sativum. Okay, so let's look at definition. So self-pollination is a process where a plant fertilizes itself without the involvement of another plant. So that is same plant. Okay, same plant. It can be different flower, same plant. So now he chose, when he started his investigation, he started by using pure breeding pea plants. Now, what is the meaning of pure breeding? Pure breeding means that plant, if I say that plant is a pure breed, that means that plant has only the gene for certain trait. That means it's not mixed. It only, let's say the plant is tall. That means that whole plant only has gene for tall. It doesn't have a mix of a short gene, okay, or gene for the other. So how do you know it's a pure breeding? We know it's a pure breeding after many generations of self-pollination. Okay, after you will know it's pure because you see, after many generation of pure breeding, you will always get the same characteristic. For example, let's say, let me give you an example. I know you don't quite understand first. Let's say I have a plant A. Okay, happens to be taller. All right, and then this plant self-pollinates. Let's say the stigma, the pollen grain of that flower drop onto its own self or another flower of the same plant. All right, it's self-pollination, and then after that, you find the next generation is also tall. After that, all right, you also self-pollinate itself, right? You do the experiment, self-pollinate, and then the next generation also tall. And then again, you repeat. Next generation, all the plants you get also tall. Okay, that means it only produces tall organisms. The plant is always tall. That means you can, after a few generations of this kind of self-pollination, you can come to the conclusion that this plant definitely must have only one type of the allele, which is the tall. It doesn't have any of the short, because if it's short, it's possible that sometimes you may skip one generation. It will appear again, after, let's say, you skip one generation, the next generation, if it has the short gene, it will appear. It could appear. So you see, after you do your self-pollination for many cycles or many generations, you find it's always tall and tall and tall. Every time you get tall. That means this plant must only have one type, which is the tall gene. So this is considered a pure breed. Confirm, right? After many generations, you cannot get any short from there, any short variation from this plant. So definitely this is, we call it pure breed. That means 100% is tulin. Okay, we call it what they call it, Parker tooling in BM lah. That means didn't mix anything, don't have a mixed breed. So say for example, let's say for example most of your, of course, your family all Chinese, right? Let's say you notice that you go trace back your family history, you find that all of your family members are all Chinese, don't have any what they call it, Caucasian or Eurasian or maybe mix of different races. You'll find that maybe in this family is all pure breed. That means it's only all the Chinese genes only. That means your grandfather or great grandfather, there were in your lineage there was no mix of any what you call maybe Eurasian blood or maybe Indian blood or maybe Malay blood or maybe what you call that the Westerners. So you see, you find that after marriage is all your eyes, or you don't have blue eyes, right? It's all brown all the way. So you can more or less you say it's all pure breed, it's all Chinese breed. Okay, your Chinese genes only lah. You don't have Indian genes, you don't have Malay genes and all that. Is we consider something like pure breed lah. Okay, it's just an idea for you to easy for you to understand. So after many generations, you don't find any different contrasting traits. You will say that this is a pure breed. Okay, so he started with pure breed first. Right, he used a pure breed plant. Sorry, where did it go? He used a pure breed plant, and then he also a tall one, and then he crossbreed with short one, a short pure breed. Okay, so you see, look here. Mendel chose pure breeding pea plants number 4 to study inheritance of several characteristics. A pure breeding plant is obtained after many generations of self-pollination. So many generations, you find it's always tall. That means these plants are called homozygous. Homozygous means the kind of gene that they have can only be one type. That means capital T for short, for tall. Okay, let's say it that way. So it's homozygous. All pure breeds are homozygous because the two we call it the two alleles there are the same type. Is homozygous. Okay, so what is monohybrid inheritance? In monohybrid inheritance, we are only concerned about studying one type of character. We don't want to see others. Let's say if you want to do monohybrid inheritance, we want to investigate on whether it's tall or short. We're not looking at the same time tall and short and also the color of the flower or the color of the seed or the color of the pot, the seed pod. We only look at one character. We just see whether the flower color on it, whether it's purple or let's say purple or white. Or we want to see whether it's tall or short on it. We don't care about all the other we call the other characteristics. We're only investigating one character at that time. Okay, that's called monohybrid. Of course, monohybrid is the easiest. Later on, we're going to learn dihybrid. Okay, so we either do monohybrid and dihybrid. Okay, we don't do more than that because it's be too complicated. So monohybrid cross is a genetic cross between two individuals that involve inheritance of one characteristic only. We are only looking at one characteristic, which is either maybe tall or short, or maybe flower color is purple or white, or maybe the seed color yellow or green. Okay, just one only. Now, so what he did, he chose the height of pea plants as a characteristic. Let's say he wants to investigate on the height. So he used, first of all, he started with, you start your investigation, you must take the pure breed, tall and short. He take one tall plant, pure breed tall, and then he crossbreed with a short pea plant. Okay, pure breed as well. Okay, he cross-pollinated them. Okay, that means what did he do? Okay, let's look at this. He took, let's say this is a pure breeding tall plant. Let's look at the diagram first. Later we look at the schematic, how to draw. Now this is a tall, this is a pure breeding, right? After many generations, he find that this is always tall. So then this one must have having pure genes. We call it pure breeding. Okay, so all tall. And then he took, let's say, the pollen grain from the flower already mature. Then he brushed it over to the other plant with this short. He took a short plant, all right? A short plant is pure breeding, all right? And then he took this pollen grain from the top plant and then he brushed it onto the stigma of the short plant, the flower of the short plant. That is called cross-pollination. Okay, so you will, fertilization and all that. Later on, you're going to get your pod. Okay, the pod will come out So this pot, let's say after it crossbreed, so wait for, of course you wait lah, right? Then you're going to get this. You are going to get flower first. After flower, then you're going to get the pot lah. This is a pot. Okay. This is a pot with the beans inside, right? The peas, not beans. Then you get this. So what happened is, now he got this. What did he do? He waited for it to become old. Don't eat lah. Of course, you can't eat it. Eat it already, no more already. You cannot. You plant it. So you wait until it becomes old. Then you take the seed. You take the seed and then you plant it. You plant it and let it grow. Then this is considered your anak, F generation. Okay. So from this plant, you got your seed pod. You take the seed. You grow it. Then only you can see, because from the seed you cannot see whether tall or short. You must give it a chance to grow, isn't it? Your seed has the genes, but it doesn't show unless you plant it. So once you plant it, you are going to see that you're going to get tall. And so happened that Mendel found that all the seeds that he took from there all became tall. There is no intermediate. Okay, in the first generation, that means the anak, the first generation, the next generation that you got, this is F1, we call it F1, we call it filial, first filial generation, F1, that means the anak, the child or offspring. The first generation, there is no short, there's no short at all. Neither is there intermediate, that means medium height. He found that all, no exception, all of them have to be tall. So he has also started wondering now, how come I crossbreed with short now, how come the short never come out? Okay, the characteristics didn't come out. Okay, never mind. He further carry on the investigation. Now this tall plant, right? All the tall plants, he self-fertilized. He do self-fertilization. Self-pollination. What is self-pollination? Again, he take the same plant, take the pollen grain, put onto the stigma of either same flower, different flower, doesn't matter, but it's on the same plant. Okay, so it will self-pollinate. So after that, it will grow pods, isn't it? So you're going to have another generation of pod. Okay, another generation of pod. So you get this right? So this is called the second generation. Okay, this is the second generation. Second generation already. So this one, but you don't grow it, you cannot see, You do not know whether they are tall or short. The seeds are there. Okay, the beans are there, the seeds are other seeds. But you do not know whether they're tall or short until you give it a chance to grow, isn't it? So you got to plant here. You plant it. And then he took all the beans, all the seeds there, and then he planted it. And he found that of all of them, he found majority of them will be tall. Okay? And there will be some, surprisingly, because some turn out to be short. Just now, the first generation, there were no short. But now the second generation, we call it F2 generation, okay? We call it second filial generation, got combination of some tall and some short, but there are no intermediate. There are no intermediate, that means there are no medium height. It's either you're tall or you're short. And so happened, he found a very interesting ratio, 3:1. Okay, what do you mean by 3:1? Let's say I have got 100 plants here. Second generation, I yielded 100 plants. Roughly out of that 100 plants, 75, roughly 75 is tall and 25 is short. So it's roughly about 3/4 of them is tall and 1/4 is short. Okay, so he got this result. And then he repeated again because, sometimes as a scientist, you have to repeat experiment in order for it to be what they call verifiable, isn't it? Your experiment has got to be what they call it. That only considered is credible, right? You got to repeat. So repeated many times. Not only using tall, he used flower color, purple and white. He used also pot color, green and yellow. He did many rounds. In fact, he has so many pea plants in his garden. And people thought he was crazy. So he repeated many times, and he always got this ratio in the second generation. Always get 3:1. Never failed. So he was very, very intrigued. Okay, how come the first generation, always get the result? First generation, no short at all. Only all tall. But the second generation, you will always get 3 to 1. The ratio is 3 to 1. Never fail. He does it again. He did it again with flower color, the color of flower. He did it again the pots. Every time, never fail. You get 3 to 1 in the second generation, and the first generation all have one type of what you call one type of characteristic, no mix. So he thought, wow, like that, that must be something. Then he stumbled upon something. Then he said, oh, this one must be a law, you know. There is a law related to this. Because it's always true. If it's always true, it always happens, that means it must be the truth. And there is a law to it. So he came up with the law, Mendel's First Law, which is the law of segregation. Okay, we go into it later. Let's teach you how to draw the diagram first. Okay, now let's look at that. Now there's no intermediate. For your first generation, F1, no intermediate. The second, also no intermediate. Intermediate means we already mix tall and short, you get medium height. No such thing. Okay, let's look at this. Now, how did he come to this conclusion? He explained it by saying that, every plant, every individual must have 2 alleles. Must have 2 alleles, cannot be one. Okay, because now it makes sense because this allele actually is in the chromosome, and you must have 2 in order for it to be a pair, isn't it? It's a homologous chromosome. One you get from the mother, one you get from the father. This is logical because you cannot have the next generation without a mother and a father. So that means your gene must be 2. One came from the father, one came from the mother. Okay, so that's what Mendel said. So this he wanted to explain why you always get that result. So the only way to explain it is by using what he has come up with. Okay, his law. So the first thing is every individual has 2 copies of the gene. That means the allele, 2 alleles. One you get from father, one you get from mother. And he also said that not all alleles are the same strength. There is one we call dominant allele, and the other one is a recessive allele. Okay, there will be not all the alleles are the same. There'll be one type called the dominant, and there's one that's recessive. Okay, that's what it means. Wait, let me check your yeah, nothing. Okay, so far nothing. Just listen first. After we go into our Google Meet, afterwards, you can ask all the questions you want. I'll explain again. Okay, so I just want for recording purposes. So here you find that this is his theory, right? Now let's see. His theory says that this is Mendels First Law. Wait. Let me go through this. See, you see here, he always get ratio roughly 3 to 1. So this is he did for height, he did for flower, he did for flower position, flower color. All this he repeated. All the 7 types of characteristics he repeated. So he did your tall and dwarf. He crossed the tall and dwarf. He crossed the purple and white. He crossed all the different contrasting traits. And then in the F1, they all show one type only. One type only. No the other type. And also no intermediate. And for the F2, he always find that there will be one characteristic which is always more. And the other one will be always less. And roughly, the ratio is like this, 3 to 1. Okay, never fail. So that means there is a law to it. And the law is called Mendel's First Law. Okay, how to explain it? He says this is called the Law of Segregation. A characteristic of an allele of an organism is determined by a pair. See, it's always a pair. That means there are 2 alleles involved. And in the formation of gamete, whenever you want to make the next generation sexual reproduction, remember we have gametes, right? So the allele will separate. So he says that in the formation of gametes, the pair of the alleles will separate. We call it segregate. Okay? Separate. It will separate. That means one gamete can only carry 1. So it only carry 1. Whether it carries the tall, one of the capital T or dominant or recessive, doesn't matter. It can only carry 1. One gamete can only carry 1. Okay? So it says here, only one allele can be present in a single gamete. Only one allele. Okay, that's what he said. That is the law of segregation. And from there, this is how he can explain why you always get 3 to 1. Okay, so now we explain using the diagram how you always get 3 to 1. Okay, let's see this way. Now let's look here. Now he used what? Remember, he used pure breed. So when he used pure breed, that means when it's tall, right? He said that tall, it could be more dominant because you see, the first F1, there are no short. That means this one must be stronger, correct or not? You see, the first generation, there are no short. You have tall, you crossbreed your tall with your short, but the first generation is you have none of it are short. That means he come to think, maybe tall is the stronger one, right? Isn't it? Maybe stronger lah. So he put that as dominant. He said tall must be dominant because they're stronger. All right. So the effect is stronger. So he said that. Now, so he put capital letter to represent tall. TT. Capital T, capital T. Okay, I write it again. Now, then the other one short. So if one is stronger, the other one cannot be also strong. So it has to be the recessive one. So he said the other one, which is the short one or the short gene, must be recessive. So he put small letter to represent. Okay, so capital T cross with small t. One is a tall plant, one is a short plant. All right, okay. Now then, during meiosis, remember when you have your formation of meiosis, you form gametes, correct? And then Mendel said, only one allele can be carried by the gamete. So you see, when you draw your gamete, right here, gamete, it takes only one. It cannot take two capital T. Okay, so the other gamete will take capital T, the other one. Okay, so there are 50%. So 50% of the gametes will be taking this allele. Another 50% will be taking this allele. Okay, so you have T, capital T, capital T. Same thing for the other, the 12th one. You will only carry one, all right, one allele, which is small t, and then the other allele also carries small t because it has no other. It only has a small t. Now, during fertilization, remember fertilization? For humans or animals, you get one sperm will fertilize an ovum, correct? One male and one female gamete. So same thing here for plants. You have the pollen grain. Any pollen grain has limitless number of chance. All right. Anything you call it random because we have thousands and thousands and millions of this pollen grain. So any one of that can fertilize any of the ovule. So you have fertilization. You take one of this gamete. Okay, I label as one now. This is one. This is two. This is three. This is four. So this is a male. Let's say this is a male. And this is a female, isn't it? Okay, you must always take a male cross with a male. You cannot be male and male cross together, right? You must be a male and a female. So you take take one of this one here, and you take another one from here. Okay, so you combine your gamete from male, take the one, you take the three from there, you combine, you are going to get what? Just add together your alleles, you're going to get capital T and small t. Okay, now leave it there first. But this is not the only chance that's possible, isn't it? Your gamete, the first one, has also equal chance to fertilize the other gamete over there. So it can also 1/4. All these lines are showing the possibility. The possibility can happen. So I put a different color. Maybe I will put, okay, I put this different color. Ooh. This one, too thick. Okay, let me put here. This is blue one. So take one of this one and you take this one. You are going to have this combination. It's still capital T, small t. Correct? These are showing all the possible combination. So this gamete here can either fertilize this or this. Okay, you have used up the possibility. Now, what about the other possibility that can happen? All right, because fertilization is a game of chance actually, right? So number 2 here. Now this gamete, you have not explored the possibility yet, isn't it? So now we try and see. Now this one, what about this one? This one can fertilize this one, 2 and 3, isn't it? So if that happens, this is the combination you're going to get. All right, this is the combination you're going to get, 2 and 3. 2 combine this gamete and that gamete. And there's another possibility, your 2 can also combine with the other gamete, right? The other one over for the short one. So you can have another possibility. So I'm going to show, let me write, okay. maybe white color. I cannot see really. Okay, so I will go to a different color. Then you can see the lines. You can see the lines clearly. Let me change this color first. Maybe I put orange. Okay, green. Yeah, green. Okay, so this one can also combine with this one, correct? You will also get, this combination. So you will see that this is the F1. F1 is the anak, the child, F1. Now, in terms of phenotype means observable. You will find that all are tall. Okay, why are they all tall? Even though they have the short gene. Remember what Mendel said, the gene can have different strength. One is dominant, one is recessive. So dominant means it's stronger. So when you have already one dominant, say for camel, you have capital T as a dominant gene, dominant allele. When you have a short one, doesn't matter because it will always show the dominant. So you can see you have one capital T, one small t, it will still be tall. So all of them will be tall in the first generation. Okay, so this one, by explaining it this way, you notice that explains his actual experiment, which he got. He got all the first, F1 is all tall. Okay, never fail. Now, then how to explain the next generation? Remember what he did with F1? He self-pollination. He took all the plants, right, and then he just picked the pollen from the plant, and then he just self-pollinate the flower, the flower itself. So he did self-pollination, right? So the next cycle he will get, the next one is, now how do you get the, you see, now the plant is TT, isn't it? All of them are like this. All of them are tall. All of them are tall, all right? So he crossbreed, right? Not crossbreed, he self-pollination. So tall, you will have, what kind of alleles will you produce? You will either produce a capital T, and because it's a small one, so another one is a small one. You can see one capital T, one gamete carries the dominant allele, the other gamete carries the recessive allele. So in this F1, it's not pure breed anymore. We don't say pure breed. You know why you don't say pure breed? Because you see inside there, the genes are not the same. You have a capital T, you have a small t. This is not a pure breed because it's a hybrid actually. The opposite of pure breed is a hybrid. Hybrid means it's a cross already, mixed already. It's not pure. Okay, 它的种不是纯, it's not 100% pure already because already mixed. Okay, now this is F1. F1 you self-pollinate. Okay, now so do over here, you also have self-pollinate. This also big T and small t. And now fertilization. Okay, after you put your pollen there over onto the stigma, you have fertilization. So what happens now? Again, the same concept. Any pollen can fertilize, has an equal chance of fertilizing any what they call any of the ovule. So you have these possibilities. First, you take this one and you combine with this, you're going to get capital T, capital T. Right? You just combine them together. Take your capital T, capital T. So if this gamete carries dominant and this one carries recessive, you are going to get you get dominant, both dominant, so dominant. Okay, now we look at the other combinations. The next one is, what about this? This gamete, this pollen has also the chance to pollinate this ovule. So now 1, 4, 1, 3, 1, 4. So this is what? You have little t, you have a small t now. Isn't it? Because this happens to carry a recessive allele. Okay, next one. What about the other possibility that you haven't explored yet? The next possibility is this one. What color did I use just now? Doesn't matter. Okay, now I use this and this. All right, take one of the male, take one of the female. Make sure it's not 1 and 2. 1 and 2 means the same sex. Female and male. 3 don't combine with 4. Because female and female. So 1 and 3, 1 and 4. 2 and 3, 2 and 4. Okay, so this one you're going to get what? You're going to get capital T and small t. Okay, and then one more combination that you have not explored yet. The other combination will be, okay, let's see the orange one. This one, 2 and 4. you will get small t and small t. Do you see that? Right? So you will see that, this one I will put here. Okay, you will see that actually you have the what you call the ratio. Let's write down the what first? Let's write down the phenotype first. Capital T is going to be tall, right? It's going to be tall. Capital T small t is also tall because I mentioned capital T is dominant. It doesn't matter you have the small, short one, the short allele, I mean the recessive one. As long as you have one dominant, it will show already. It will show the tallness. So the second one here, the third one here is also tall. Okay, because this is capital T small t. And here, you see the difference here? Now your short comes out. That's why you see, so it is so amazing. Now, this next generation, you will get a short, or we call dwarf lah. And the ratio is, this is 1, this is 2, you have 3. You will have 3 tall and 1 dwarf. And it happens all the time because this is a law. Whatever you do with the other types of these what you call characteristics, you will still get 3 to 1. Okay, but 3 is not all at the same time. Only one is the homozygous dominant. The other 2 are actually heterozygous. Okay, so like this. How do I draw it? Okay, like this. Capital T, small t. This is 1. You have 2 of this, and you have 1 of this one. Okay, so this is homozygous dominant. This is heterozygous. You have 2 of it. And then you will have homozygous recessive. Okay, so you will have, that's why in the phenol F2, you will come out as if, wow, short come out again. All right, from the parent, you have short. Then the F1, no short at all. Then the F2, wow, the short come out again. Okay, it appears. So it's because of this law. This is the only way to explain. There's no other way. So I would say Mendel is pretty smart, very smart in order to come up with this thing. Because otherwise you cannot explain. You cannot explain why you have 3 to 1. Okay, there's no other way to explain it. Okay, now besides drawing this, you can also use what we call a diagram called a Punnett square. We call a Punnett square. Sorry, it ran away. 1 minute. Sorry, it's a Punnett square to show the result of the crossing. Okay, how do you use a Punnett square? Okay, very simple. You put all the gametes into the table. So first generation, F1. Now, F1, what kind of gametes do you have? This is F1. F1, you have this. You have this, right? You have this combination. 2 capital T and 2 small t, right? You have this one. 2 capital T, 2 small t. So what you do is you fill up the table here, horizontally or vertically. You have 2 capital T here. You have 2 small t here. What you need to do is you just have to combine. Pretty much like just what we did. 1, 3, 1, 4. 2, 3, 2, 4. Okay, you do 1, 3, and 1, 4. You get 2, 3, 2, 4. Now, that's how you combine it. If you don't want to do that, you can also do Punnett square. So you take this one and this one, you combine together, you get capital T, small t. You also combine this one and this one, you combine together, you get capital T and small t. Okay, so you combine the top here, capital T, capital T, capital T, small t, capital T, small t. So you get all tall plants. Okay, next one. What about the second generation? Okay, look at the second generation. What kind of alleles do you have in the second generation? You have this combination already. You have this combination. Capital T, small t, capital T, small t, isn't it? They are not all capital T, not all small t. So capital T, small t, capital T, small t. So you put it here. Capital T, small t, capital T, small t. So you just need to combine. Okay, so combine this one, take this one and this one, add together, you get capital T, capital T. All right, then, right, and then you combine here and here, you will get capital T, small t. And then next, you combine, let me just rub this away. Okay, you combine this and this, you get capital T, small t. And lastly, you combine that one and this one, you get small t and small t. All right, and then you will get this is tall, this is tall, this is also tall in phenotype, and this happens to be the only one that is short or dwarf. Okay, 3 to 1. All right, understand? Now I want to show you, to warn you about one mistake that students always make. All right, you know, when they draw their table, you know what they do? I'll just start on an empty plate here. Wait, sorry. I'll start on an empty slate here. Okay, I'll do like this. All right, this is how you draw. All right, tall. I'm going to draw from parental generation to the F1 generation to the F2 generation. Okay, let me write down. Parental. Now always write all this at the side, because in SPM, it was marked last time. If you just draw, you never explain all this, you never draw all this, you get it wrong. So you got to write here parental tall. And then you cross with short. Before that, you always write your what they call key. Capital T is dominant, capital T is dominant allele for, this is how you do it. Okay, now follow me. Dominant allele for tall. Always write your key first. You're telling people what is the symbol for, right? This one right away. Okay, small t is recessive allele. I'm going to start from scratch. So please listen. This is how you answer questions for short or dwarf. Put short. Okay, short is better. Okay, so you start with the key here first. Then you draw your parental generation. Parental generation. Okay, so this is the phenotype. Okay, phenotype. Phenotype is you show what is it, use what is it, tall, is it short. Okay, then what about the genotype? Now you got to write a genotype. Genotype is capital T, and small t, because they are pure breed. You start off with pure breed. Okay, both are pure breed. They are homozygous dominant, homozygous recessive. Okay, now the next generation, the next thing you need to do is to show your what? Meiosis. So here, split your arrow here. This one shows formation of gametes. So you do like this. So you write here, meiosis. You have to write this, otherwise I tell you what's the difference. You have to write this is to show meiosis, all right? And this is to show gametes. So you put a capital T here, small t, small t. Okay, next you got to show fertilization. Now, fertilization is very simple. Remember, 1, 3, 1, 4, 2, 3, 2, 4. Always follow this rule. You know why I tell you to remember this? Because I've got students previously who did not know how to do. You just simply draw like this. Very beautiful. How do I do it? Okay, wait, is it like this one? No, it's like this. Did it very nicely. I can tell you she did it very nicely. Oh, yeah. Very symmetrical, isn't it? Very nice, right? Oh, another way is she did like this. It was so nice. How was it? Is it this way? Oh, yes. That's even beautiful. All right. Now tell me, is that right or wrong? Tell me. Let me see. So beautiful. So symmetrical. Okay, now what happened? He took number 1 and, okay, let's say 1, 2, 3, 4, or Sheila. Okay, 1, 2, 3, 4. 1 and 3 combined. This is 1, 3. So you got, this is 1, 3. Then he went to combine 1, 3 again. Combine here, see, 1, 3. Then he used 2, 4. And combine 2, 4 again. You repeatedly, the person did repeated this 1, 3 again, 1, 3 again. Where is your 2, 3? Where is your 1, 4? It looks beautiful, right? It looks so fantastically nice. This is wrong. You don't draw like this, okay? Because you have repeated your combination. 1, 3. This is again 1, 3. It looks nice, but once I see if it's so nice like this, it's definitely wrong. It has to be 1, 3, 1, 4, 2, 3, 2, 4. You have 4 different combinations. You should not be repeating the combination. This is wrong. Okay, it looks so nice. So you don't do this, please. You'll get it wrong. You'll get it wrong. So please write fertilization here first before you start. Fertilization. All right, fertilization. And you do like this, 1, 3. Yes. Write it down. Before you forget, write it down, because you're going to get confused when all the lines come. If you finish drawing all the lines and then all you draw all the T, then you will get confused. You will get it wrong, definitely. So 1, 3, draw. Then 1, 4, draw. Capital T. Okay. Then 2, 3, draw. And 2, 4, draw. Okay, 1, 3, 1, 4, 2, 3, 2, 4. Okay, now this is your F1 generation. Write down this at the side. Okay, this is your genotype. What you see is genotype, but you can write your phenotype as well. You can put here tall, 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 tall. These are the phenotype. Phenotype is outward appearance. Okay, now this is the first crossbreed. Now you're going to self-pollinate. You take any two. You take any two of this F1. Now your F1 is going to self-pollinate. Take your F1. You're going to self-pollinate because all of them are the same. Take your F1, self-pollinate. What happens here is you're going to have again the same process, formation of what? Formation of gamete. So here, don't forget F1 generation. Here is meiosis. You write all that. You don't write here on the left-hand side. It's all wrong. If you just write all the circle, and the lines, you get it 0. Okay. So you got to write what is this gamete. All right. Gamete like this, and put in your capital T, small t, capital T, small t. Okay. Next is you got to show your fertilization between the gametes. So you write here fertilization. Okay. So what you do is always do single one by one. 1, 3. Okay, draw first. Don't go and do all the lines. You will combine wrongly. Capital T, capital T. Then 1, 4. Capital T, small t. Then 2, 3. Capital T. Sorry. Okay, what did I get? Capital T, capital 3. Yes, small t. Capital T and small t. And 2, 4. Small t, small t. So here, write down F2 generation. Okay, follow up. And then you can write your phenotype here. This is already the genotype. All the symbols are already the genotype. So you write here your tall, 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 and short. So here your phenotypic ratio. If the question asks you for phenotypic ratio, you put 3 tall, 3 tall, 2 dots. 2 dots here, ratio, always 2 dots. 3 talls, 1 short. If the question asks you for genotypic ratio, genotypic ratio, how you gonna write it? You can't put all 3 together for the genotypic. You need to put 1 is capital T, capital T. Then you have 2 that is capital T, small t. You have 1 that is small t, small t. Always remember genotype, you need to use symbols. For phenotype, you use words. Okay, so this is how you answer the question. You remember you have to write what's on the left-hand side here. Where is it? Okay, here. You have to remember to write this. Otherwise, it's wrong. You'll be penalized if you don't write this. Okay, a lot of people just write whatever is on here, the drawings on it, wrong. Okay, now, so let's check whether you understand, and you take a few time for you to take a picture. All right, now this is talking about a crossover using the same law of Mendel's First Law of Inheritance, which is called Law of Segregation. Law of Segregation. You make sure whenever you draw it, your gamete must only carry 1 allele. Okay, and then you use that law to explain what is the what they call the next generation, the characteristic of the next generation. We can also talk about human ability to roll the tongue is known to be dominant. Roll the tongue, the U shape. Okay, I show you. I can. So I must have the dominant allele inside my cells. Okay, inside my chromosome. I must have it. So it's otherwise I cannot roll it. Okay, the inability, they can't do it, they can't roll it no matter how hard they try. They want to learn it, you can't learn it, you cannot roll it. Okay, you can try yourself whether you can roll, you cannot roll, look yourself in the mirror. Okay, then you look at this picture. Now try to complete the diagram here. What is the phenotypic, the genotype and of F1, the next generation? Okay, it doesn't go to F2, it just go to F1 only. Okay, you try this. Afterwards, you discuss this. And for those who are not in my class, I mean not in my students, I will show the answer to this in the next lesson. Okay, so this is today's lesson. I just wanted to teach you how to draw the monohybrid inheritance. Okay, so this one you can just read through. It's the same thing, right? It just shows you the same thing. It's explaining the monohybrid. Just take a picture and then you can look at it later. All right. Okay. So that's it for today. We will go to Google Meet, and then any questions you please ask until you understand, because it can be quite confusing at first. Okay. So I will see you in a while. All right. For those out there, all right, I will say bye-bye now, and we will meet you in another lesson. Okay. Bye-bye.